Yeah. Dear students, today we will be discussing about methods of gene transfer, and these are in continuation with the yesterday lecture that is about the cloning and recombinant DNA technology. Once you have made the recombinant plasmids, the next target is to transfer those recombinant plasmids back into the biological systems or biological cells, which could be bacteria, which could be yeast, fungi, or human cell or animal cell, for their expression. And for transferring those recombinant plasmids or vectors back into the living cells, we need different techniques. And today we'll be discussing those methods which are used to transfer those genes back into the living cells. So today the lecture would be entailing, I have to divide this lecture because this presentation is a bit long. I would be dividing this lecture into two parts. One would be the entailing with the viral vectors, and other would be the entailing with the physical and different chemical methods which could be used for gene transfer. So in this two lectures, we will be actually identifying the two main methods of gene transfer, and then we will be comparing the different viral vectors which could be used for this purpose, and uh, which of these viral vectors is good for a specific therapeutic purpose. Then we will be tomorrow's lecture, we will be discussing about the physical and chemical methods of gene transfer, their comparison of different nanocarriers used for gene delivery, as well as we will appreciate the potential of non-viral methods as a therapeutic strategy for gene therapy. So moving further, what is gene transfer? So if we come up to the gene transfer is, uh, uh, we have given different, scientists have given different terminologies to these gene transfer methods. If we introduce genetic material into a bacteria, we simply call it as a transformation. Means introduction of any recombinant genetic material back into a bacteria is such a thing is called transformation. Moving further is that if we we do a transfection. Transfection means introduction of genetic material into a eukaryotic cell. Means that could be fungi, any plant, animal, yeast, whatsoever a eukaryotic cell is. If we are transferring a recombinant DNA material back into a eukaryotic cell, such a thing is called transfection. While if we use viruses for transferring or introduction of genetic material back into any of the eukaryotic or prokaryotic cells, such a thing is called transfection. While there is another technique which is called lipofaction. In this, we introduce the genetic material using liposomes. So these are the different gene transfer methods or terminologies which are used based upon the cell type. So moving further, there are two types of gene transfers. One is the stable and other is the transient gene transfer. Stable gene transfer means that you have a stable gene expression for a long period, and transient gene transfer means that you don't have a stable uh, expression of the gene, rather you have a transient gene expression for a smaller time period. Stable gene transfer could be defined as or could be achieved by plasmid integration into the host genome, or episomal expression or replication of the transferred plasmid. Means that that whatever plasmid or recombinant DNA you have transferred back into the cell, either it becomes part of the nuclear DNA, or on the other hand, it expressed as an extra chromosomal part which have a capacity, autonomy to replicate itself, having an independent origin of replication array. So episomal expression and replication is actually also considered as a stable gene expression or stable gene transfer. While when we move towards transient gene transfer, it simply could be the transfer of foreign DNA in which the foreign DNA or the recombinant DNA does not integrate into the nuclear genome. Rather, it would be degraded or diluted through mitosis or cell divisions. Whenever with the passage of time, this transient expression would lose, and we could not have a long-lasting effects of those transformed recombinant DNA particles. So how the plasmid integration takes place. This is the example of a stable gene transfer in which we added a plasmid, and that plasmid integrates into the nuclear DNA, and once this cell replicates, the plasmid DNA along with the nuclear DNA replicates, and both the cells have the same type of nuclear material in it, and it's a stable integration of the uh, genome plasmid which you have added or transferred, and that becomes integrated into the nuclear genome. This is called this is again it's not a example of episomal vector with again it could be classified under the stable gene transformation in which these plasmids have an independent origin of replication, and they replicate along with the nuclear DNA when the cell replicates, and both the cells, daughter cells, have the plasmid with it. While another third condition is, which is actually the transient gene expression, and in this the plasmid is not replicated independently, autonomously, and the two daughter cells don't have the same type of genetic material, nuclear or episomal DNA. So this type of gene transfer is transient expression of the transgene, and you will not have a long-lasting effects. Now coming to the delivery methods, that you have made the clone or a recombinant vector, and now you have to deliver that vector back into a living cell. That living cell could be both prokaryotic origin, eukaryotic origin, or anything which we think that is of importance. So there are two methods, delivery method. One is the viral method, another is the non-viral method. Viral methods, in viral method, we used different types of viruses for this, and we will go by all these details of these all viruses one by one later. And while in non-viral procedures, we have some physical methods and some chemical methods. These all non-viral portion will be discussed in the next lecture. So moving further, that what viruses are. What viruses are, actually, we all know it. These are non-living entities, but they could replicate and multiply within a living cell using their own. They carry a genetic material within themselves. That genetic material could be both DNA or RNA, and they are named based on their names, genetic material they carry, as well as they have a membrane or envelope or capsid on it. These are made of fatty acids or proteins, and they are on these coatings. They have ligands, which are actually proteins, which creates a capacity to the that you actually provide the capacity to the virus to attach to the different living cells. So what I could do is kindly, boys and girls, kindly remove these annotations which you have done this. This creates a lot more problem. Okay, so moving further, if we after viruses, they have the capacity and ligands to these ligands help the viruses to attach to the living cells, and we will be discussing all this later, that how we could do it. Now moving how viruses work. So normally we all know it, viruses, you have studied it many times, that viruses replicate through both lytic and lysogenic cycles. Here they have explained the lytic cycle, that viruses enters the body of the cell, and after entering into the cell, uh, that actually enters the, and that virus is later on stripped off its things. Its coating is stripped off by the lysozymes and all the stuff, and the nucleic acid of the virus become released from the capsid of the virus, and that nucleic acid of the virus then enters into the nucleus of the cell, which it has entered, and start controlling the things, and it start dictating the things to generate their own proteins of which are actually required by the viruses to replicate and synthesize more viruses, and later on these all viruses are released by destroying the cell. So this is how it works, and how viruses replicate within the living cell. So moving further, if we consider of our viral vectors, what could be the general requirements? So we need such a viral vector which is actually deficient in replication, or it should have a controlled spreading in vivo. It should not replicate tremendously within the living cell if it enters into the cell. So moreover, it does not, that viral vector should not have undesirable properties of or pathogenicity, that it should cause some disease within the cell. So we have normally scientists what do is they actually remove the Rex gene as well as the pathogenicity virulence genes from the out of the Vir gene out of these viruses to make them desirable for transformation and transcription work. The viral genome should be capable of to accommodate the therapeutic gene. Means those viruses should be capable to accommodate small or big genes within them, which are actually the transgenes, which need to be transferred into the other cells. So coming to the delivery methods, the viral vectors includes, we have adenoviruses, which could be used. We have adeno-associated viruses, which are also named as parvoviruses. We have herpes viruses, retrovirus, and lentivirus. These are 5 different viruses which are used now for this purpose. And now we would go to all these viruses one by one to study what are the benefits and what are the disadvantages of these viruses. And in the later on, we will be having a comparison of all those. So starting with adenoviruses, these are big viruses of almost size of 70 to 100 nanometer, and these are double-stranded viruses. These are recombinant vectors. We call them as recombinant vectors because certain these adenoviruses could cause diseases. So scientists have removed many crucial genes out of them and such after removing those crucial genes, which could be disease-causing genes, we call them as recombinant vectors because now they could not cause any disease to them, so we call them as recombinant vectors. These adenoviruses vectors are actually replication deficient. If we come to the advantages, they have a higher titer, means whenever you are actually, we generate these viruses in the lab. When I say generate these viruses in the lab, means that we actually generate viruses, or we pack these viruses. In other words, we pack these viruses in the lab on other mammalian cell lines. We will be discussing all these packing. Packing means you generate such a virus which has your own transgene to transfer back into the other cells. So this packing mechanism is done on a mammalian cell, and when this packing is done, we achieve low titer or high titer, that we are successful in getting more viruses or less viruses. If we have more viruses, the titer would be high, and if we are not successful in having packing in viruses at a low quantity, we have a less titer. So normally these adenoviruses have a high titer. They could affect a very huge quantity of species of organisms, means they could the spectrum of to infect many organisms, from human cells to animal cells to many other eukaryotic and prokaryotic cells. So their spectrum of species to infect or transduce is higher. Again, within the organisms, they could target a wide range of cells, from kidney cells to pulmonary cells to heart cells to neural cells. They could infect a wide range of cells. The other advantage is they have a high transduction efficiency, means their transduction efficiency is much more. They could transduce a lot more transgene into the cells. Lastly, they could transduce the primary and non-dividing cells, means the cells which are not dividing, they could even be transduced via these adenoviruses. The biggest two disadvantages of these are that these adenoviruses don't have a stable gene expression, rather they have a transient gene expression. The simple reason is they are not after transduction, the viruses are not successful in transducing the plasmid DNA into the nuclear DNA. So that's why they have a transient gene expression. And other is the immunogenicity of due to their a bit larger size. Next is Adeno-Associated viruses or Parvovirus. Again, you see the structure. These are small viruses, almost 40 to 50 nanometer size, and they are single-stranded viruses. They could be divided into wild type and the recombinant types of these adeno-associated viruses. The wild types are actually normally these adeno-associated viruses are not reported to be disease-causing, and that's why in some experiments scientists have used the wild type. But the biggest benefit of this adeno-associated virus is the site-specific integration in the host cells. Normally, adeno-associated viruses integrate the transgene in the human cells at chromosome 19 at a specific locus, that is AAVS1. AAVS1 locus, this is present at the chromosome 19, and normally adeno-associated viruses integrate the transgene into the nuclear DNA of humans at chromosome 19. So this is one of the biggest advantage of this adeno-associated viruses. And they have the transduction capacity, again, both on the proliferating and non-proliferating cells, means dividing and non-dividing cells. And we have a possible chromosomal integration. These are the benefits. But at the same time, there are few disadvantages with them, that is immunogenicity and the because it's a single-stranded DNA virus, they have the possibility to mutate. I told it that Y type is, because it's not a big virus, it's a small virus. So the capacity to accommodate the transgene in this virus is small. It could accommodate from 4.5 to 4.9 kb or almost 5 kb of transgene in it. It could not accommodate much more bigger genes in it. So for that reasons, this is also one of the pitfall of this adeno-associated viruses, that it could not accommodate large genes in it. Now moving further to the herpes virus. Again, this is a big virus. It's almost 200 nanometer of size with a huge genome, and it's again a double-stranded virus. Again, recombinant vector, because many of the genes have been, which are pathogenicity or replication genes, are removed from this virus by the scientists. So that's why we call it as a recombinant vector, and they are certainly deficient in replication properties. So advantages of these viruses is that they have the capacity to transduce the non-dividing cells, and the biggest advantage is that they have an affinity towards neuronal cells or neuronal tissues, means nerves, neurons could be more easily transduced by using these herpes viruses as compared to the other viruses. And the biggest, because of its size, it could accommodate large fragments of foreign DNA or transgene, that it could even accommodate almost 40 to 50 kb of transgene in it. But the biggest disadvantage of this herpes virus is the transient expression. Means you could not achieve a stable expression. The reason behind is the transgene which you are transducing using this herpes virus would not get integrated into the nuclear genome. So there is always a transient expression with the herpes virus. Going further towards retroviruses, from the word retro means it contains RNA, and it's a single-stranded RNA virus. The advantage is that its host range is too wide. It could infect or transduce in multiple organisms and multiple cells type. And the advantage is its stable integration with the host genome, that it actually, the transgene is being integrated into the host nuclear DNA. But certainly when you talk of integration into the host genome, that means that it's not specific in terms of retroviruses as it was specific in terms of adeno-associated viruses, that we were knowing that normally adeno-associated viruses integrate the transgene at chromosome 19 at AAVS site or locus in the human genome. But in terms of retrovirus, it is not like that. It is non-specific integration in the genome, and so there are chances that you could have different cells. So certainly when you are doing a transaction with the viruses, you need a selection markers to move on and do select certain cell type which are normal, healthy, and all that. The advantage with this retrovirus is that you cannot achieve high titer of viral stocks when you are doing packing of these viruses. And when you do this packing of viruses doesn't achieve high titers. They have limited working towards proliferating cells, means they could only affect the proliferating and dividing cells. Non-proliferating or non-dividing cells could not be transduced with these retroviruses. And the other problem is their oncogenic properties, means tumor formation properties of these retroviruses also is amongst one of the pitfall of this retroviruses. Retrovirus structure, it's simple that retroviruses enters the cell by fusion and entry. Their capsid and all this is removed. They enters into the nucleus where they integrate into the nuclear DNA. They start controlling and preparing, replicating their own DNA and proteins, and then this transcription would assemble at the cell membrane, and budding takes place, and the mature viruses are released into the medium or the cellular space to infect other cells. So moving towards the last but not the least is the lentiviruses. These are subclass of retroviruses, and again they are single-stranded RNA viruses, and they are mostly used nowadays for gene therapy and all type of stuff for this purpose. You can see the structure that there is an RNA structure with a capsid and matrix, and then there are different ligand binding proteins at the surface. So advantages of this is stable integration with the host genome, means that they could give a stable gene transfer capacity. Ability to integrate into the genome of non-dividing cells even, means the retroviruses don't have that capacity to integrate or transduce the non-dividing cell, but these lentiviruses have the capacity to both integrate into the genome of both dividing and non-dividing cells. Less tendency towards oncogenesis, but few cases are reported in which there is a tumor formation possibilities. So moving further, last but not the least, that how we do lentivirus packaging and replications. This is the biggest, and all viruses are packed in the similar manner. We do it on a mammalian cell line. Normally used mammalian cell line is HEC293 or HEC293 T cells. We culture all those cells, and then we expose those cells with three plasmids, one carrying the transgene, other carrying the genetic material for the viruses, and last but not the one which is actually expressing the proteins, capsid, and all that of that viruses. When all these three plasmids are transfected to the cells, they would certainly replicate on those mammalian cells, HEC293 T cells, and they will produce their own genomic material, packing cells. And what would happen that these cells, these genetic materials, and all these things would generate and pack a virus, and this virus would be literally released into the medium. So once this is released into the medium, this was actually established that you would have a high titer or low titer of this. If you have a lot more viruses into the medium, and these viruses would be later used to do a transduction experiment. So we actually purify these viruses from the medium in which we have cultured HEC293 T cells, and use these viruses for transducing doing a transduction experiment of the transgene in which we are interested. Moving further, how these normally cells enter the cells. These viruses enter the cells. So there are non-alveolar viruses normally work through receptor-mediated endocytosis, that they attach to the certain receptors, such as adenoviruses and the adeno-associated viruses attached to specific receptors, and then endocytosis takes place, or in terms of plants, pinocytosis takes place. While few viruses which are actually enveloped viruses, such as retro or lentiviruses, they attach to specific receptors, and they are actually they move through membrane fusion. That membranes are fused, and then the material is transferred back into the cells. So endocytosis and receptor mediated could be divided into three types endocytosis. One is phagocytosis, that simply cell membrane engulfs the solid particle. Other is that we have material in the extracellular fluid, and pinocytosis takes place and vesicles are formed. While last is the receptor mediated endocytosis, in which the particles are available in the extracellular fluid, and they attach to a specific receptor, and then endocytosis takes place and coated vesicles are formed, and then the genetic material is transferred into the cytoplasm. And from the cytoplasm, lysosomes degrade it, and the genetic material is released, and that moves to the nucleus. Now, making a comparison of all what we have discussed about the viruses, in a tabular form, if you see it, that we have adenoviruses, adeno-associated viruses, herpes simplex virus, retrovirus, and lentiviruses. The particle size of adenoviruses is almost 70 to 100 nanometer. Adeno-associated virus is 20 to 25, 150 to 100, 100. Cloning capacity means what amount of transgene they could accommodate within itself is adenoviruses could accommodate almost 8 to 10 kb of fragment. Adeno-associated virus almost 4.9 to 5 kb. You can say it. Yeah, here you see this herpes simplex virus could accommodate almost 40 to 50 kb of transgene. So a big amount of genes could be accommodated using this herpes simplex virus. Retroviruses and lentivirus 8 and 9 kb. Chromosomal integration is possible with only adeno-associated viruses, retrovirus, and lentivirus, while HSV and adenoviruses doesn't show the capacity to of stable integration or stable gene transfer. Now coming to the transduction unit per ml means vector yield, which is actually the titer when you are actually packing the viruses in any mammalian cell. So we could have high titers with both adenoviruses and adeno-associated viruses, while we have a moderate titer with the retroviruses and lentiviruses. Means when you are doing a packing of these retroviruses, lentiviruses, we don't achieve much higher titer. We don't have no much packed viruses to be used for further transduction experiment. If we talk of entry mechanism, normally these two are working through receptor-mediated endocytosis. Uh, Means adenoviruses and adeno-associated viruses work using by receptor-mediated endocytosis, while these three actually enter the cells by membrane fusion. If we come to the transgene expression, adenoviruses have very short uh, transgene expression of peaks, while adeno-associated viruses have could go up to months. Similar is the case with the HSV. While in terms of retroviruses and lentiviruses, we could have transgene expression for years. Now coming to the oncolytic potential, adeno-associated viruses doesn't have, and retroviruses doesn't have this oncolytic potential, but adenoviruses, retroviruses, and lentivirus has this potential. And uh, if we go to the oncogenic potential, firstly doesn't have it, but retroviruses and lentiviruses could create tumors. So we have to be very careful with them. Now coming to the risk of replication in vivo, uh, in terms of HSV and retroviruses, it's a possibility of concern because they could be disease causing. While in terms of AAV, we all know it that is not a big deal because they doesn't till now they nothing is reported that they could cause disease. So it's not a concern for that either they are replicating or not. So for today, we have discussed till the viruses, and these viruses could be used for gene transfer in other living cells, both prokaryotic and eukaryotic. And in the tomorrow's lecture, we will be considering about the next half, that is about the physical methods which could be used to transfer genetic material into the cells. So I hope so you have understood today's lecture, and we will be having a fruitful discussion tomorrow at the same time. Stay blessed.